Hello everyone, my name is Vi and I'm from the Graduate School of Medicine at Songkun University. Today I'd like to present our study which demonstrates that ablations of PMT1 in endothelial cell cause endothelial dysfunction and aggravate COPD attributable to dysregulated NF-kappa signaling. This slide shows the general structure of blood vessel. Both artery and vein share a similar architecture. The innermost layer is made up of endothelial cell. The middle layer consists of smooth muscle cell, and the outer layer is composed of collagen fiber. On the other hand, the capillary consists of a single layer of endothelial cell. Endothelial cells play a crucial role in gas and nutrient exchange, regulations of vascular tone, immune response, vascular homeostasis, and angiogenesis. Under healthy conditions, endothelial cell functions as a barrier to maintain tissue homeostasis. However, when endothelial cell function is impaired, it can contribute to a variety of diseases, including retinopathy, lung disease, sepsis, stroke, and so on. The target genes in our study is protein arginine methyltransferase 1 or PMT1. It belongs to PMT-THY1 family and is known as a critical enzyme that can catalyze the symmetric methylations on arginine residues of target protein. These modifications are important in cell proliferation, differentiation, and other cellular processes. Interestingly, scientists have found an elevated level of ADMA, mainly synthesized by PMT1 in cardiovascular disease, and this ADMA can inhibit nitric oxide production and can lead to endothelial dysfunction. However, until now, the specific functions of PMT1 in endothelial cells remain unclear. To explore this, we generated an inducible endothelial cell-specific PMT1 knockout mouse. In these knockout mice develop severe pulmonary hemorrhage and significantly increased lung weight. Besides that, we also observe a left ventricle dysfunction, disrupted endothelial junctions, and elevated cell death in the lung tissues. Additionally, transcriptomic analysis of knockout lung shows significant upregulation of genes related to inflammation. Using single-cell RNA sequencing, we further analyze the pulmonary endothelial cell and identify 11 distinct subpopulations. In the knockout lung, the proportions of GCAP4 subpopulation was significantly increased. We also found that the GCAP4 were associated with elevated expression of pro-inflammatory genes and the activation of NF-kappa signaling. To validate this, we inhibit PMT1 in HUVEC cells using furamidine. As expected, we found increased NF-kappa activity in furamidine treatment. IP experiments also confirm that PMT1 binds and methylates P65 subunits of NF-kappa, suggesting a direct regulatory mechanism. COPD is a chronic lung disease characterized by a progressive airflow limitation and persistent inflammation. Endothelial functions is known to play an important role in this disease, but so far there is no drug that can really treat COPD. Because we observe a strong phenotype of inflammation in the PMT1 knockout mice, we decided to look more closely at how endothelial cell PMT1 is involved in COPD. To explore PMT1 roles in this context, we analyzed single-cell RNA sequencing data and found that PMT1 is highly expressed in pulmonary endothelial cells, but its expression is significantly reduced in the COPD mouse model. Also, in our elastase-induced COPD mouse model, we confirmed the reduction of both PMT1 and ADMA level in the lung endothelial cell. In a COPD, endothelial cell-specific PMT1 knockout mice exhibit more severe lung damage, such as increased capillary enlargement, cell death, and cell senescence in the lung tissues. Also, in vitro, PMT1 inhibition by furamidine enhanced TNF-alpha-induced NF-kappa activation in HUVEC, suggesting NF-kappa B dysregulation may involve in endothelial cell senescence. We then overexpress PMT1 in human cell, and we observe that PMT1 overexpression mitigate NF-kappa B-induced endothelial cell senescence. Finally, to test this in vivo, we use an AAV vector to overexpress PMT1 specifically in endothelial cell of COPD mouse, and consistently, the PMT1 overexpression improve the lung function and reduce NF-kappa B activation in our COPD model. In conclusion, our findings show that PMT1 is essential for maintaining endothelial cell function by regulating NF-kappa B signaling. Loss of PMT1 leads to endothelial inflammation and promotes COPD pathogenesis, and the results highlight PMT1 as a potential therapeutic target for COPD treatment. Our study was recently published in Advanced Science, and if you are interested in learning more, please feel free to look up our paper. Finally, I would like to thank my supervisor, Professor Jong-sun Gang, and all the lab members for their invaluable support. Thank you for your listening.